Welcome to my channel Deep Learning Biology. My name is Dr. Vikas Bhardwaj. In this video, I am going to teach you about proteins, the primary structure of proteins. So be with me and learn clearly about primary structure of proteins. Now try to see here. You must be knowing, if you squeeze the cell, what you will get? 70% water. And here, if you do analysis, percentage weightage of different biomolecules within the cell: lipids 2%, carbohydrates 3%, nucleic acid 7%, 1% DNA, 6% RNA, and proteins 15%. Now, from this data, you can conclude that the largest mass fraction of cell, it is going to have proteins. Now here I just want to tell you, if you see, you must be knowing proteins, they are polymer of amino acids. In a protein, what you are going to see, amino acids, they have been joined together by peptide bonds. Here what you are going to have, polypeptides. Many amino acids, they have been joined together by peptide bonds. That's why we are going to say polypeptides. Remember, proteins, they are the workhorses in a cell. All the tasks of your body, all the tasks of your cell, they are being carried out by proteins. Try to think here. Some of the proteins, they are going to act like enzymes. Some of the proteins, they are going to act like hormones. Some of the proteins, they are going to act like structural elements. Some of the proteins, they are going to act like signal receptors or transporters. Here you see the various activities which are occurring within your cell, within your body. It is just because of proteins. That's why we are saying proteins are working horse of the cell. Now here I just want to tell you, if you see the scientist, we want to study, we want to know the function of proteins which are present in our body, or the proteins which are present in different organisms. Now here, to know the function of proteins, generally scientists, what they study? They study the structure of the proteins. Try to see here. You must be seeing various objects around you. By looking into the structure, yes, you can easily predict the function. So here the scientists, what they actually do? They study structure of proteins so that they can know what is the function of that protein. Most of the times, scientists initially what they are going to do? They crystallize the protein. After getting the crystals, by using suitable techniques, instruments like X-ray diffraction, they are going to study the structure of proteins. Yes, in this process they are going to use computer. Okay, this way they are going to figure out what is the structure of the proteins. Now here you must be knowing protein structure. It is generally studied at different level. One is primary structure, second is secondary structure, third is tertiary structure, and the fourth is quaternary structure. Today in this video, I am going to teach you about primary structure of the protein. So we are going to have discussion about the primary structure of protein. Yes, in upcoming videos, we will have discussion related to secondary, tertiary, and quaternary structure of proteins. Now here, if you see the primary structure of the protein, it is going to tell you, okay, what kind of amino acids they are being present within the protein. Hopefully you know, it is clear in your mind that protein, they are polymer of amino acids. And keep in mind, generally 20 or 22 amino acids, they are being found within the proteins. Now here I just want to tell you that if you see these amino acids which are present only in the proteins, these are known as proteinogenic amino acid. Keep in mind 22 amino acids, they are generally found only in proteins, and these amino acids, they are known as proteinogenic amino acids. Yes, there are also non-proteinogenic amino acids. There are also non-proteinogenic amino acids. Now here keep in mind the primary structure of the protein. It is going to tell you about the amino acid sequence. What kind of amino acid sequence are present within the protein? Yes, primary structure of the protein, it is also going to tell you about covalent bonds, generally the peptide bonds and the disulfide linkages which are present within the proteins. So here, by studying the primary structure, generally what we get, we came to know about the amino acid sequence which is present within the protein and peptide bonds, disulfide linkages which are present within the proteins. Now here, if you see the amino acids, okay, generally you must be knowing they are going to have amino group and the carboxyl group. Yes, they are also going to have side R group except glycine. Now here you are going to have a polypeptide. Let's suppose here you are going to have amino acids A1, A2, A3, A1, A2, A5. They are being joined together by peptide bonds. Now try to see here. You are going to have free amino group. At one end you are going to have free amino group. This end, it is known as N-terminal end. Now here you see in between each amino acid residue, what we are going to see, formation of peptide bond, and finally at the end you are going to have the COH group, carboxylic group, it is C-terminal end. Now keep in mind, whenever we are going to read any protein sequence, we are going to read it from N-terminal to C-terminal, from N-terminal to C-terminal. Now let us suppose you are going to have two amino acids A1 and A2. Let's suppose there is formation of peptide bond in between A1 and A2. What will be? There will be removal of water. Okay. So I have shown here in a simple, I have shared here in a simplified way. Okay. It is a complex process. We will learn it when we are going to discuss the process of protein synthesis. Okay. So here in between A1 and A2, what you are going to see, formation of peptide bond. Now here you are going to see the formation of dipeptide. Keep in mind 20 different amino acids. 20 different amino acids, they are going to join together in different way in different proteins. Now here these amino acids, if you are going to calculate the average molecular weight of the amino acids which are present within the proteins, it will come out 128. Now here you see, with the formation of peptide bond, there is removal of water molecule. Okay, water molecule, it is going to have molecular weight 18. You minus this. Okay, so it will come out 110. Okay, so here you have to keep in mind this one, average molecular weight of each amino acid residue within the protein is 110. This information is very much useful. Now try to see here, by using this information you can calculate the number of amino acid if you have been provided the molecular weight. Let's suppose there is a protein, its molecular weight is 1,10,000 Dalton. Now you can easily calculate the number of amino acid. Very simple. Just you divide 1,10,000 by 110. Okay. So it will come out 1,000. So here you can easily find the number of amino acid in a protein if you have been provided the molecular weight. Just you have to divide by 110. Yes. In another way, if someone has given you number of amino acid and someone is going to ask you what is the molecular weight, can you predict? Just multiply it by 110. You will get to know about the molecular weight. So hopefully it is simple, clear. Now let's move forward. Okay. So here keep in mind that each protein, try to see within the cell you are going to have millions of proteins, and each protein is going to have distinct number of amino acid sequence. There, keep in mind each protein is going to have distinct number of amino acid residue. Yes, each protein is going to have distinct sequence of amino acid residue. Okay, so here, okay, you can see that each protein, the different proteins, they are going to have distinct number of amino acid residue. They are going to have distinct sequence of amino acid residue. Now let us move forward. Okay, remember the proteins which are going to have different function. The proteins which are going to have different function, yes, they are also going to differ in amino acid sequence. Yes, here keep in mind functionally similar proteins. The proteins which are going to have similar function, they are also going to have similar amino acid sequence. The proteins which are going to have similar function, they are also going to have similar amino acid sequence. Now let us see here that within a protein, what you are going to see, certain amino acid sequence, they can tell you more about that protein. Yes, certain amino acid sequence within a protein, they can tell you about cellular location of the protein, where that protein can be present, whether it is present in nucleus, cytosol, or it is exported out of the cell. So within the primary structure of the protein, what you are going to get to know about amino acid sequence, and few of the amino acid sequence within the protein, they can tell you what can be the cellular location of the protein. Now here, yes, certain amino acid sequence of the protein, they can also tell you what kind of chemical modification can occur within the protein, whether there is going to be attachment of sugar group at some particular amino acid sequence. Okay, for example, in glycoproteins, what you are going to see, attachment of sugar residue at some particular amino acids within the protein. So the primary structure can easily tell you now what kind of chemical modification that protein can go. Same way, yes, in sometimes there is attachment of lipid groups. It can lead to formation of lipoproteins. At what point lipids group might attach to the protein, at what amino acid sequence the lipid group might attach, that information you can get from the amino acid sequence. So certain amino acid sequence, they can tell you about cellular location, they can tell you about chemical modification. Yes, some of the sequence, they can also tell you about half-life of the protein. Every protein is going to have life. Okay, now by studying certain amino acid sequence, you can also predict the half-life of the protein. Now finally, I just want to tell you, you can also study the evolutionary history of life by studying the primary structure of the protein. You can get to know about molecular evolution of closely related proteins. Okay, and this way you can predict the evolutionary history of life if you are going to study closely related proteins. Okay, this way you can study the process of evolution. Now try to see here. Here you must be knowing within the eukaryote you are going to have one protein EF1 alpha. In prokaryotes, bacteria, you are going to have another protein EFT. Now these two proteins, okay, they actually carry out similar function. They are involved in synthesis of proteins. They are involved in synthesis of proteins. And what you will get to know, that these two different proteins present in two different organisms, they are going to have similar sequence. So here keep in mind, the proteins which are similar in sequence, generally they are going to be similar in function. So the proteins having similar sequence, they are going to have similar function. They are going to belong to same family. Yes, such type of proteins, they are known as homologs, or you can say homologous proteins. The proteins which are similar in sequence, similar in function, belonging to same family, they are homologs or homologous protein. Now try to see here. If you see in the case of homologous protein, yes, these homologous protein, they might be present in same species. They might be present in same species. In this condition, they are known as paralogs. Confused? Don't worry, I will make it clear. Let's suppose there are two different proteins, A and B. These are two different proteins. They are having same sequence. They are going to have same function. And let's suppose they are present in mouse. They are present in same species. These are homologous, present in same species. Now these A and B protein, they will be known as paralogs. Homologs present in same species, they are known as paralogs. Now there can be another situation also. Here if you see, let's suppose you are going to have two proteins, they are homologs and they are present in different species. Let's suppose here you are going to have X. This X protein is present in mouse. Then you are going to have Y protein. It is present, let's suppose, in human being. These are two homolog. Okay, they are similar in sequence, but they are present in different species. So these proteins, they will be known as orthologs. So homologs, they are homologs present in same species, they are known as paralogs. The homologs present in different species, they are known as orthologs. Hopefully all the points, they are clear to you. Now let us move forward. Try to see here the bioinformaticians, okay, by using various bioinformatics tools, okay, they can actually study the evolution of different types of homologs. Okay, so change in amino acid sequence of homologs proteins in one species, in another species, and this way they can study the evolution of life. Okay, the evolution of life on earth. Okay, so this is about the primary structure of protein. Now let us discuss finally the primary structure of protein, insulin, the insulin hormone. Try to see here, you are going to have two chain, A chain and B chain, and here you can see the peptide bond, or this one disulfide linkage. So here this is disulfide linkage between cysteine residue, okay, within the protein. Generally what you are going to have, disulfide linkage. So here you can see there are 3 points where you are going to see formation of disulfide linkage. Yes, disulfide linkage, they give stability to the proteins. Now here you see there are two chains. One is A chain. Within the A chain you are going to have 20 amino acid residue, 21 amino acid residue, while in the B chain you are going to have something like 30 amino acid residue. So within the B you are going to have 30 amino acid residue. So here if you see in the insulin, you are going to have total 51 amino acid residue. Here you are going to have two chain, okay, A chain and B chain. So hopefully you have learned clearly about primary structure of proteins. Thank you for watching this. Please do like, share the video and subscribe to my channel Deep Learning Biology. Keep learning, keep watching Deep Learning Biology.